Hello and welcome to the MU 418 lecture about macrophage polarization and recruitment. So I'll be talking about how macrophages can be polarized to different phenotypes to promote or inhibit cancer. And in this image on the left, it's electron microscopy of two macrophages. Both are on these myofibroblasts, the activated fibroblasts, which are massive, by the way. But the macrophages, they phenotypically look different when they're polarized to their M1 phenotype to their M2 phenotype, as demonstrated by this image. So to overview this lecture, I'll be talking about macrophages, their normal function, and when they switch phenotypes. And these phenotypes are known as M1 and M2, and they can be identified by different phenotypic markers. And I'll also talk about flow cytometry analysis. I'll also cover some strategies to target the tumor-associated macrophages, or TAMs. The objectives for this class are to understand the phenotypic switch of macrophages between the M1 and M2 phenotypes and how it can affect cancer progression, and to understand the cell surface markers of M1 and M2 macrophages and how they are used in research. The resources that we have on Canvas, we've got the Cancer Immunology textbook. There's a chapter about macrophage polarization there. If you want further reading, there is a video on iBiology that's also available on Canvas, talking about very in detailed description of flow cytometry. So to begin with, macrophages, they are actually a differentiated monocyte. So monocytes in the blood, they migrate and leave the circulation, differentiate and become macrophages. They're mainly involved in the innate immune system response, where they phagocytose dead cells, bacteria, foreign particles, and they're involved in antigen presentation. So in this small diagram, you can see that migration in response to different growth factors causes the monocyte to migrate to the site where it's needed, and then differentiates in response to other growth factors to become a macrophage. So this is the, I guess, simplified version of M1 and M2 macrophages. I think a lot of immunologists would argue for a long time that, within the M2 macrophage, there are multiple other subtypes. But for the purpose of this course and this lecture, I'm going to group all the M2 macrophages together, subtypes. So when you have a differentiated macrophage or a monocyte, it can phenotypically switch between M1 and M2 phenotypes, depending on the stimulants. So in response to TLR ligands and LPS, we've got TNF alpha, tumor necrosis factor alpha, or interferon gamma. These are your inflammatory, infection signaling, typical signaling cytokines and growth factors that will push the macrophage towards an M1 antimicrobial phenotype, which is anti-tumor as well. It's high energy, but it uses a glycolytic metabolism, and we get increased production of inflammatory molecules, and so we've got a lot of intracellular reactive oxygen species, and accumulation of arginine to generate nitric oxide to really just attack. This is like the attack macrophage, let's say. But in response to IL-4 and IL-13 are some examples of stimulating interleukins that can drive M2 macrophage expression. This figure's a little funny now that I see it. It looks like this is a really happy macrophage with mitochondrion for eyes, but it's in normal conditions, I guess it's a happy macrophage. It's not the attack macrophage. It's the healing macrophage. But in cancer, this is the enemy, unfortunately. So it has a higher metabolic output and uses a different metabolic pathway to the M1. It doesn't generate anti-inflammatory, it doesn't generate inflammatory intracellular reactive oxygen species. It is producing anti-inflammatory cytokines to promote modeling of the extracellular matrix in terms of like wound healing and promotion of angiogenesis. It is mainly in normal is the healing tissue repair macrophage. So macrophage polarization, it happens in normal conditions, but in cancer it is a kind of corrupted for tumor progression. So macrophages are recruited into a tumor. They would be ordinarily in response to growth factors and chemokines, and you would expect that a macrophage would be there to shut down the tumor. Fortunately, it switches to the M2 phenotype to promote angiogenesis and healing, mainly by growth factors, colony stimulating factor 1, CSF1, and by the CC motif ligand 2, CCL2. These are the two most well documented or well studied cytokines and growth factors that are associated with recruiting and stimulating M2 type macrophages. So tumor-associated macrophages, TAMs, are involved in tumor promotion in a number of ways. So they produce growth factors for tumor cells, they promote angiogenesis, they can remodel the extracellular matrix to promote invasion, and they also have a strong immunosuppressive function. So using this nice figure here, you can see these processes in more detail. So macrophages promote and express cytokines and growth factors to basically enhance the tumor growth. So epidermal growth factor, platelet-derived growth factor, and fibroblast growth factor are all cytokines involved in tumor growth, as well as angiogenesis and anti-inflammation. In promoting angiogenesis, TAMs can release interleukins and other factors like IL-8, particularly VEGF, which promotes angiogenesis and growth of endothelial cells and migration of endothelial cells towards the tumour. They are also involved in remodeling the extracellular matrix. So proteases, such as the matrix metalloproteases, are released by macrophages, the TAMs, to degrade the surrounding tissue that might be restricting growth of the tumour and allows tumour cells to metastasize and invade. And finally, immunosuppression. They have a very well documented role in anti-inflammatory and immunosuppressive functions. So macrophages, TAMs, can upregulate the programmed death ligand 1, PDL1 marker, which is an anti-T cell, it's a T cell inhibitory marker, and they're increased on TAMs. They can increase the expression of other anti-inflammatory cytokines and signaling molecules, but also they can in fact attract and recruit the T cells, the T regulatory and T helper cells that can also perpetuate the M2 phenotype and recruit more monocytes to get an even more anti-inflammatory, immunosuppressive tumor microenvironment. So obviously there are major differences between the M1 and M2 types, and it's important to identify the M2 types within a tumor. And to do this, we can look at the expression of markers. Thankfully, well, as much as their phenotypes differ, so do their cell surface markers. Here is a non-exhaustive list of cell surface markers, cytokines, and other markers. Immunologists would attack me probably for this. I'm not a purist. So these are the most well-known or the most well-documented. It doesn't represent an exhaustive list. Don't write a thesis on it, but this will get you by in a flow cytometry experiment, but not in front of a panel of immunologists. But you can see that some very basic markers are shared between both M1 and M2 macrophages. So this you would use to identify what macrophages you have in total, and some other markers you can use to exclude other cell types from, say, a batch of whole blood that you get. The toll-like receptors, those are upregulated generally in response to infection, so on. So they're on the M1, they're activated in M1 phenotype macrophages. And then a couple of these special, CD means cluster of differentiation, I should say. These CD 163 and 206 are arguably exclusively found on the M2 type. So that's a good way to differentiate your M2 type. In terms of secreted factors, actually I put this point on here when I thought it might be a live class to invite discussion of how these markers could be useful, but I guess I'm just giving you the answers. So the cell surface markers can be used for antibody-directed things such as flow cytometry and so on. The secreted factors could be measured by ELISA. So again, anti-inflammatory versus pro-inflammatory and recruitment of other immune cell types. And then some other markers that, again, arginase and MMPs matrix metalloproteases are involved in the extracellular matrix remodeling and degradation, and INOS is intracellular nitrous oxides, again for the pro-inflammatory and antimicrobial defense. So I'm going to give a overview introduction about flow cytometry analysis, because I'm not sure at the differing levels of experience with flow cytometry in this class. I mean, feel free to skip this if you know a lot more about flow cytometry than I probably do, but this is a really basic overview of how flow cytometry works, basically. So when you're looking at flow cytometry, you're using it to analyze the expression of markers. Predominantly, it's cell surface markers, but you can also you can look at intracellular markers, and it's used to characterize many different cell types within a heterogeneous population. So in this diagram here, you're putting your sample in, and you've got here basically blue and red cells, but you might have up to 8 or 9 different cell types, many more, all going through the flow cytometer. So your cell sample is a suspension, either from your cell culture experiment, some tissue, or whole blood. You can, any variety of sample is prepared in a particular way to analyze on the flow cytometer. And you study your proteins of interest using fluorochrome labeled antibodies to label your particular marker for analysis. So when the cells run through the cytometer, you have a big heterogeneous sample going up through the nozzle, but when cells are run through the cytometer, there's this high pressure is applied to suspension to create a single cell stream of fluid. So there's a laser light source is detecting your fluorochrome labeled antibodies, but the cells must pass the laser one cell at a time. So it's a really high throughput way to analyze a huge heterogeneous population of cells, but it's only as good as your antibodies. So as each cell, single cell, passes through the laser, they might have a single color, they might have multiple colors of fluorochromes, but there are several detectors within the instrument to measure the forward and side scatter of each individual cell. That's for cell size and shape, which can also be used to differentiate your cell types, as well as the fluorescence detectors for your antibody labeled cells. So here, it's a little bit blurry, my apologies. This purple round thing is a cell, I think that's the nucleus there. So this blue triangle is being picked up by this antibody with the red label here. The laser's hitting it, it's emitting light, and it's being detected in this case by the PE laser. And it also has I guess that's a green color, detecting a different marker. So this could be, CD45 and CD206 or similar. And in the green channel, the laser excites it at a different wavelength, so it's detected on a different detector. So in this case, FITSI. Okay, so you can get multiple information about a single cell out of your entire population. Simultaneous multi-parameter analysis of each cell within your population. So when you run a flow cytometry analysis, here is a typical scatter plot that you would get out of a flow cytometry analysis of, I believe it was a whole blood. Yes, I think it was a whole blood analysis. I took this from a paper where they demonstrated their gating strategy to identify macrophages and specifically tumor-associated macrophages. So you always start with your forward and side scatter. They're not fluorochrome labeled. They're size and shape. So you're looking for, again, if you have different size cells that you're looking for within a population, especially whole blood, you will get some really small populations, and then you might get larger populations, but generally you want your cells to be within this middle range. So if you've got forward scatter one side and side scatter on another axis here, a round cell is going to be the same size, right? Arbitrary level 1 and 1, it should sit in the middle. So that's your main population. Next, you will then exclude any cells that have stuck together. You only want to analyze single cells, otherwise it'll skew your analysis. So again, if you're using an arbitrary little plot here and you've got forward scatter here, forward scatter height versus the area. If the area is larger than the height, it's because you've got doublets. So you only want this 1-to-1 ratio. And then same again, you can use side scatter height versus area. Again, if you've got a larger height than you have area, you've got doublets, but it's in a different conformation. So once you have your size, population, and you've excluded any doublets, you're looking at your fluorochromes. So first, they gated on, they wanted to look at all cells that were expressing CD45. All leukocytes express CD45. And this can be a way to actually exclude leukocytes as well. You can negatively select if you don't want to look at any leukocytes and you only want to look at a different cell type out of a tissue, then you can also use it to negatively select. But in this case, obviously, they wanted the CD45, which is expressed by all macrophages. They also here included a live dead stain. In this case, the dead cells take up the stain because their membrane is not intact, so they just absorb the dye. So here on this this population down in the corner, see if I can get my laser pointer back. It's being really strange. Right. So these are log scale axes. The highest population here is CD45 positive. So they took that population. They also wanted the lowest absorbance of the dye that they used to look for dead cells. So all of these cells here, up to the 10 to 3, are the live cells, but they want the live and CD45 positive. Whereas these might be CD45 positive, more likely they're dead cells and debris that have autofluoresced. But importantly, they took up the dead stain, so they're excluded. So you've got living leukocytes. From this population, you can look at your other markers. So within those cells that express CD45 and they're alive, then you want to look at laser pointer. So then they looked at excluding, as I said, you can use markers to exclude from your analysis as well. They wanted to only look at leukocytes that were basically not T cells and B cells. So this time they didn't need a counter stain. They just wanted to look at a single stain. So CD3, CD19, CD56, this kind of combo stain will get rid of, you can select out your T cells and B cells. So this population here, between 10 to the 3, 10 to the 4, is their T cells and B cells from their sample. And so we want the negative population. The forward scatter, again, here doesn't really matter. It's just a way to see you've got more than one population here. Next, from this negative population, they wanted to identify, from this negative population, then they could finally look at macrophages. So using these two markers, CD11B and CD14, macrophages express both. So again, laser pointer. You can see this population here is negative for both. It doesn't express either. There's a sort of intermediate population that might be undifferentiated macrophages, maybe monocytes. There could be, undecided cells. But this population here, very clearly, is a dense number of cells. This is the macrophage population. And then finally, they didn't have an image for this. It would have looked similar to this graph. Of this macrophages, they sorted for the CD163 positive. They used CD163 as their marker for their tumor-associated macrophages in this study. I know that was laborious, but I want, in your assessment, you to perform a gating strategy on an experiment. So I'll talk about that at the end, but it's a lot to take in, I know. So when it comes to targeting tumor-associated macrophages, they seem like a good idea to target them, and it's just challenging. So there are two broad mechanisms in order to target TAMs for anti-tumor activity. One is to kill the TAMs, try and kill them specifically, or inhibit their recruitment to the tumor. The second broad aim is to reprogram them, turn off M2 and switch them to M1 to try and promote cancer killing. So this image that I took from a paper describes that kind of nicely in a summary. Strategies to do so, but you'll see it's not quite that easy. So the first strategy to kill or inhibit the tissue tumor-associated macrophages, we can either inhibit the cytokines that recruit them to the tumor, so using the CCL2 as a target, and these are CCL2 recruits TAMs and promotes phenotype switching. So anti-CCL2 antibodies are in phase 1 or 2 trials in multiple cancers. Limited success so far. So to deplete TAMs, there are some agents, but they're limited by their specificity against the M2 or TAMs. In research, you can use CSF1 receptor inhibitors. So by targeting CSF1, that is present on all macrophages. So it's not specific. Neither are bisphosphonates. And cloudinated liposomes are a funny thing. They've got a nice cartoon. So you have a liposome that's filled with cloudinate. A macrophage, due to its phagocytic nature, is attracted to this liposome to phagocytose it, but it dies. So none of these are specific strategies to target the M2 macrophages, because we still want the M1 macrophages. There are some early preclinical studies about targeted antibodies against M2 specific markers, including CD163 and CD206, and conjugating these with drugs to try and improve their efficacy. But again, the expression of these is not necessarily ubiquitous or limited to M2 macrophages. So not every M2 macrophage will express these, but it's under investigation. So the other mechanism is to reprogram the TAMs and switch their phenotype back towards an anti-tumor M1 phenotype. So TLRs, toll-like receptor agonists, are trialed, and one is approved to try and activate the inflammatory foreign particle antibacterial response to target tumors. So there are several TLRs that could be targeted, and kind of counterintuitively, TLR2 and 4 are expressed on the surface, but TLR3 and 7 exist within the endosome, which is intracellularly, and they have elicited the best response in experiments and trials. However, there is one approved. As I said, it is a TLR7 agonist, and it's for topical treatment only, for squamous cell carcinoma and basal cell carcinoma. It's known as imiquimod. Poly-IC is an agonist for TLR3, one of these endosomal TLRs, and it has been shown to have good activity and can promote an M1 response, but the formulation of particularly trying to target an endosomal receptor is a challenge. So again, there's also antibodies to try and promote the phagocytic nature of macrophages. I talk about this in the antibodies lecture, but CD47 is a inhibitory marker expressed by cells to signal to macrophages to not eat them. So by inhibiting CD47 on cancer cells, they use CD47 to hide from macrophages, but antibodies against CD47 could increase the phagocytic activity of macrophages against cancer cells. And these are in early phase trials. And there are also some inhibitors being tested and developed preclinically, specific inhibitors that are being also trialed, preclinically, experimentally trialed, to try and switch the phenotype of macrophages and improve their pro-inflammatory responses. So to summarize, I discussed macrophage phenotype switching, the differences between M1 and M2 phenotype macrophages and how they have distinct roles in normal, non-cancer, innate immune responses, as well as in cancer progression. I also showed that TAMs can promote tumor growth, neoangiogenesis, extracellular matrix remodeling for invasion, and can be immunosuppressive. So they truly are the enemy. Cell surface markers on macrophages, they can be used to identify and phenotype different macrophage types by flow cytometry, and there are many iterations of cell surface markers to identify macrophages and other cell types. And there are limited but ongoing investigations and strategies in order to target these TAMs to disrupt cancer progression in different phases, and their aims are to either kill TAMs, to inhibit their recruitment, or to try and reprogram them to switch them towards an M1 pro-inflammatory phenotype. So as I alluded to, I have assessment based around flow cytometry and cell surface marker expression. So I'll be giving some flow cytometry results, some scatter plots with different cell surface markers, and I want you to physically draw, physically or electronically draw gates around each population and name and describe these populations according to what cell surface markers were used. So if you have questions or if it's confusing or any other concerns, please email me. But thank you for your attention. See you next time.